Hello friends, today we will discuss about basics of statistical tests. Hello friends, now in this video we are going to learn about the hypothesis testing process. Now first thing is we need to consider question. The population mean age is 50. Now we need to sample the population and we are going to find sample mean. So we will set our null hypothesis like mu is equal to 50 and alternative hypothesis that mu is not equal to 50. So first thing is suppose sample mean age was x is equal to 30. So this is significantly lower than claimed mean population age of 50. Suppose if null hypothesis is true, then probability of getting different sample mean would be very small. So we need to reject null hypothesis. In other words, we can say that getting sample mean 30 is so unlikely if the population mean was 50. So what we can conclude from this? So we can say that population mean must not be 50. Now we will see this sampling distribution of x by normal distribution curve. Say for example, we are considering mu is equal to 50. So we are considering if h0 is true, when in fact this were population mean. Suppose we are considering mu is 50, that is mean of population is 50, then we are considering h0 is true. Suppose we are saying that our sample mean is 30. Then if it is unlikely that you would get x of this value, x is mean of this value as 30, then we need to reject null hypothesis. Now we will discuss another important thing. Say for example, if sample mean is close to stated population mean, it means null hypothesis is not rejected. But if sample mean is far from stated population mean, then null hypothesis is rejected. Null hypothesis is rejected means we are going to accept alternative hypothesis. But what is the meaning of far here? So how far is far enough to reject null hypothesis? Say for example, we are considering critical value of test statistics which create line in the sand. So for decision making, we need to answer some question like how far is far enough. So main part in hypothesis testing process is statistical test. So the statistical test uses the data obtained from sample to make decision about our population. So whether our null hypothesis should be rejected or not, that is rejected or accepted. We are going to consider test statistics. So this is numerical value obtained from statistical. Next thing is critical region. So it is region of rejecting null hypothesis. Next important term is critical values. So to separate critical region and acceptance regions, we are using critical. We will consider our terminologies in graphical way, or we will consider normal distribution curve for better understanding. So this is sampling distribution of the test statistics. So you can see here critical values denoted by green lines and star points. So two regions we are considering here, region of acceptance and region of rejection. So region of rejection is marked by red line and that smiley denotes region of acceptance. So we are considering critical values for rejection and acceptance of our test. Then too far away from mean of sample distribution is considered in region of rejection. So we need to reject our null hypothesis if our values falls in region of rejection. So for considering critical values, we are considering p-value. So p-value is always probability of obtaining test statistics. which might be equal or more extreme than observed sample value given, that is null hypothesis is true. So this is also known as observed level of significance.